Hello everyone. Let's get started. Today we're going to start our discussion about the macromolecules. Remember, there are 4 macromolecules that are found in living cells: protein, carbohydrate, nucleic acid, and lipid. Today we're going to be talking about carbohydrate and lipid. The way we'll approach this topic is by first discussing carbohydrates and the roles that they play in living cells. We'll discuss some nomenclature or vocabulary that we use when we're discussing carbohydrates. We'll discuss the general types of chemical reactions that produce carbohydrates and break down carbohydrates. These are known as hydrolysis and dehydration reactions. We'll talk about what joins individual units of carbohydrate together, a covalent bond called a glycosidic linkage. We'll discuss the role of 2 particular types of carbohydrates, and those are oligosaccharides and polysaccharides. When we discuss the lipids, we'll look at the 3 different types of lipids and how they differ structurally. We'll look at the type of covalent bond that builds lipid, and that's called an ester linkage. And we'll finish up by discussing the roles that lipids play in cells. One of the things that makes a discussion of carbohydrates a little bit difficult is that there are terms that are in common use in the human language that mean different things than the way those terms are used in science. For example, in science, a carbohydrate is also called a sugar or a saccharide. In common language, though, a sugar is sucrose or table sugar. So it's very confusing sometimes when scientists discuss sugars because most people think of white sugar when they hear that term. It turns out that carbohydrates or sugars or saccharides are quite common in living cells. Things like glucose and fructose and galactose are abundant. In fact, carbohydrates are absolutely essential for cells to survive. They are the molecule that cells use to make ATP for energy, and they're also used to build other larger molecules. When we look at the term carbohydrate, it actually translates to carbon hydrate or carbon water. What this name tells us is that inside of all carbohydrates, we find 3 of our 4 key elements: carbon, hydrogen, and oxygen. If you break down the structure of a carbohydrate, you'll actually find that there are carbon water units. Some number of these carbon water units in the different types of carbohydrate. If you take a look at the drawing at the bottom of the slide, you're looking at 3 different carbohydrates: glucose, galactose, and fructose. These 3 carbohydrates are isomers. And what that term refers to is the fact that they all have the same chemical formula. They all have the same number of carbons, hydrogens, and oxygens. But there are slight structural differences. That's what makes each one of them its own unique carbohydrate. If you look at the glucose molecule, for example, this is a strange way for us to think of glucose. It's a strange way for us to draw glucose because normally glucose exists as a ringed structure. But it's helpful to look at the linear structure because it allows us to compare the positioning of certain functional groups. When you look at the glucose drawn this way, you can see that on this carbon, there's a hydroxyl group, and it's drawn off to the right-hand side. If you compare that to the galactose molecule, that same hydroxyl group is drawn over to the left-hand side. In other words, when these molecules are in 3 dimensions, the orientation of that particular hydroxyl group varies between glucose and galactose. It's in a different position. If we look at fructose, we see another change. The hydroxyl group on that carbon looks to be in the same position as it was in glucose. But if we travel up the molecule to this carbon and the final carbon, that's where we see the difference. Remember when we were talking about functional groups, we looked at a carbonyl group, and we said that there are 2 forms that the carbonyl group can take. The carbonyl group can sit at the end of the molecule, or it can sit in the interior of the molecule. In fructose, the carbonyl group is interior. In galactose and glucose, it's at the end. Again, if you added up the carbons and the hydrogens and the oxygens, you find the same number in these 3 carbohydrates. It's their structural chemistry that differs. Carbohydrates play a list of functions within a living cell. As I said earlier, it's carbohydrate that is the substrate for the production of ATP, and it's the ATP molecule that cells use for energy. A single carbohydrate, glucose, is the most important of the carbohydrates when we talk about energy production. Carbohydrates are used for the storage of energy in different types of organisms. For example, in plants, glucose is stored as a polysaccharide called starch. There's a similar molecule in animals called glycogen that again serves as an energy storage molecule. Carbohydrates also play an important role in structural components of cells. Molecules like cellulose and chitin and peptidoglycan are all structural carbohydrates. We'll talk about each one of those in a few minutes. Carbohydrates are important because they sometimes provide building blocks for larger molecules. And a good example of this is nucleic acid. Inside of both DNA and RNA, there is sugar, there is carbohydrate. And finally, carbohydrates play an important role in cell identity. There are both glycoproteins and glycolipids that are found on the surface of membranes. And it's these carbohydrate portions of the proteins, carbohydrate portions of the lipids, that give a cell its own unique identity. Let's take a minute and talk about the nomenclature that we use when we're discussing different varieties of carbohydrate. Remember, the terms carbohydrate, sugar, and saccharide all mean the same thing. There are different sized carbohydrates in cells, though, so we need different terminology for each one. The simplest form that a carbohydrate can take is called a monosaccharide. In other words, one, that's what mono means, one saccharide or one sugar. Something like glucose or galactose or fructose would be considered a monosaccharide. These are also sometimes referred to as simple sugars. If we take 2 monosaccharides and we join them together covalently, we create what's called a disaccharide, di meaning 2. Disaccharides, such as lactose or milk sugar, which is made of 1 molecule of glucose bound to 1 molecule of galactose. Disaccharides are also sometimes called simple sugars. Simple sugars are relatively easy to digest. Next, we get to oligosaccharides. Oligo translates as few. So these are molecules that have few saccharides in them, or few sugars. And that means between 3 and 20 individual sugar molecules bound together. And finally, we have the polysaccharides. Poly means many. These are the largest types of carbohydrates. They contain 20 or more of the monosaccharides bound together. One thing to note about the nomenclature around carbohydrate, anytime you see a term that ends in OSE, that's a signal that that molecule is a carbohydrate. Glucose, galactose, fructose, lactose, sucrose, maltose, those are all carbohydrates. So carbohydrates can exist as single monosaccharides or in combinations that are covalently bound together. When carbohydrates are built in the cell, the general type of chemical reaction that does this work is called a dehydration reaction. The other term that you'll read in reference books is condensation reaction. I tend to use the term dehydration myself. When you bring two monosaccharides together side by side, and now we're drawing these in their ring formation, what's going to happen is we're going to find a hydroxyl group sticking off the number 1 carbon on this ring that aligns with a hydroxyl group sticking off of the number 4 carbon on this ring. Remember, all of the carbons in a ring structure are numbered. Carbon 1, carbon 2, carbon 3, carbon 4, and then an oxygen at the apex. These two hydroxyl groups are reactive, and in the presence of an enzyme, they will come together and a molecule of water will be released. Hence the word dehydration. We're losing water when these two molecules come together and covalently bind. If you move over to the other side of the reaction, you can see that covalent bond between the glucose on the left and the fructose on the right. This is an unusual looking covalent bond because it has an oxygen in the center. This is what we refer to as a linkage. It's still covalent, but it's different from what we ordinarily think of as a covalent bond. This type of linkage that joins two monosaccharides gets a name. It's called a glycosidic linkage. So just like any chemical reaction in a living cell, the formation of a glycosidic linkage requires an enzyme. When we look at different carbohydrates, one of the things that we see is different forms of the glycosidic linkage. These are referred to as either alpha or beta forms, and they differ by how that glycosidic linkage is oriented in relation to the rest of the molecule. For example, in an alpha 1,4 glycosidic linkage, and these numbers 1 and 4 describe the two carbons that are involved, where the hydroxyl groups are located. In an alpha 1,4 glycosidic linkage, the oxygen in the center of the linkage is actually oriented away from the rest of the molecule. And what that means is that covalent linkage is more exposed. That means when the time comes to break that glycosidic linkage, The enzyme that does that job can get access fairly easily. Carbohydrates that contain alpha-1,4 glycosidic linkages are therefore more easy to break down. They're easier to digest, in other words. In comparison, a beta-1,4 glycosidic linkage has the oxygen in the center of the linkage, oriented toward the rest of the molecule. This makes the linkage less exposed, harder for the enzyme to get access to, and therefore harder to break, harder to digest. If you look at the two pictures at the bottom of the slide, over on the right-hand side, we can see an alpha-1,4 glycosidic linkage. This molecule on the left represents glucose, and we have a second glucose on the right. This is the number 1 carbon. This is the number 4 carbon. Each one of those carbons used to have a hydroxyl group coming off of it. The two hydroxyl groups, in the presence of an enzyme, came together and formed the linkage, releasing a molecule of water. Notice how this linkage is drawn. The linkage is drawn projecting away from the rest of the molecule. That's the alpha configuration. We find alpha-1,4 glycosidic linkages in carbohydrates such as starch and glycogen. Remember, these are storage forms of glucose, and it's a way for the cell to store energy. It makes sense that both starch and glycogen are easy to digest. In other words, the cell has access to that glucose when it needs it. Over on the left-hand side, we have a beta-1,4 glycosidic linkage. Again, two molecules of glucose are drawn, and the linkage is drawn between the number 1 carbon from the glucose on the left and the number 4 carbon from the glucose on the right. Notice how the linkage is drawn. This time the oxygen is oriented closer to the rest of the molecule. This makes the linkage harder for the enzyme to get access to, therefore harder to break apart. Beta-1,4 glycosidic linkages are found in carbohydrates like cellulose. Cellulose is not intended to be a storage form for energy. Cellulose is a structural carbohydrate. So it makes sense that this molecule would be harder to break apart. When the time comes to break apart a carbohydrate, the general kind of chemical reaction that does this work is called a hydrolysis reaction. That word hydrolysis translates as water breaking. So in comparison to the dehydration reaction, where a molecule of water gets released, in a hydrolysis reaction, a molecule of water gets consumed. And that's what they're showing us in the diagram. Up at the top of the diagram, we can see a disaccharide, two simple monosaccharides that are joined together with a glycosidic linkage. When an enzyme is available, and we need a different enzyme for each type of carbohydrate, that enzyme, in the presence of water, is going to break apart the glycosidic linkage that joins these two carbohydrates. What we're going to find is a return of the hydroxyl group attached to the number 1 carbon on this molecule, which is glucose, and the number 4 carbon on this one, which is fructose. I want to say just a few words about oligosaccharides. Remember, an oligosaccharide is a carbohydrate that has between 3 and 20 monosaccharides within it. Oligosaccharides are used by cells in order to identify themselves. They're generally found attached to either protein or lipid in the outer surface of the plasma membrane. Over here on the right-hand side, the lower right-hand side, we can see a drawing of a section of membrane. You can see the individual phospholipids drawn in gray. This red structure right here represents a protein, and attached to that protein is this branched structure. This represents a glycoprotein. Over here we can see another carbohydrate. This time it's attached to one of the phospholipids. This would be called a glycolipid, and this would be called a glycoprotein. Inside an organism like a human being, there are cells within the immune system that are constantly on the alert, looking for any cellular material that doesn't belong inside that organism. And the way the immune cells can figure that out is by reading these carbohydrate tags. Cells called sentinel cells come along and will essentially evaluate the carbohydrate oligosaccharides that are present on the surface, the outer surface of cells. If the oligosaccharide marks the cell as being part of the body, the immune cells will ignore it. If they read the oligosaccharide as not belonging in the body, the immune cells will attack and destroy it. The other purpose of these oligosaccharides is in cell-to-cell signaling. These are carbohydrates that allow cells to communicate with each other. The polysaccharides are the most complex of the carbohydrates. Remember, a polysaccharide has at least 20 monosaccharides within it, all covalently bound together with glycosidic linkages. Polysaccharides serve two purposes inside of cells. They can be energy storage molecules, and they can be structural support. We're going to learn about 5 individual types of polysaccharide: glycogen, starch, cellulose, chitin, and peptidoglycan. Glycogen and starch are energy storage forms of polysaccharide. Glycogen is the form that's found in animals. Starch is the form that's found in plants. Cellulose, chitin, and peptidoglycan are structural support polysaccharides. Cellulose provides the structural support in plants. It helps form the cell wall. Chitin provides structural support in both insects and fungi, as well as other organisms. Chitin is what makes up, for example, the exoskeleton in certain insects. When you step on an insect and you hear it crunch, that's the chitin in its exoskeleton. Finally, there's peptidoglycan, which makes up the cell wall around bacterial cells. We're going to take a few minutes now and look at how these individual polysaccharides are built. We'll start with glycogen. Glycogen, because its purpose is energy storage, is a polysaccharide that's made entirely of glucose. And the glucose molecules are joined together in a way that makes them easy to break apart with an enzyme. When you look at glycogen, you're going to see long chains of individual glucose molecules bound together with alpha-1,4 glycosidic linkages. One of the things that makes glycogen structurally distinct is the fact that the long chains of glucose molecules don't stay in a linear form. They twist into helices. Those helices, those linear helices, are then branched periodically, and you end up with something that looks like this. The reason that glycogen is branched like this is because it allows more individual units of glucose to be stored in a smaller space overall. More glucose can be packed in to glycogen this way. Now, when it comes to the branches that we find in glycogen, we're going to see them coming off of the helices approximately every 10 glucose units. So there'll be a branch, and then if we count 10 glucose units over, there'll be another branch, and so on and so on. These branches come off perpendicular to the rest of the linear helix. And that means that we're going to need a slightly different alpha glycosidic linkage. If you look at the drawing at the top on the right, you can see this. Here's a glucose molecule. Here's an alpha-1,4 glycosidic linkage. Here's another glucose, alpha-1,4. Here's another glucose, and so on. If we look at this glucose, we can see that there's a branch coming off of it with another chain of glucose molecules. In order for this branch to come off perpendicular like this, we can't have an alpha-1,4 linkage. We need a slightly different conformation, and that is alpha-1,6. So it's the first carbon and the number 6 carbon that are joined together, again with one of these alpha glycosidic linkages. When we look at starch, it has a lot of structural similarity to glycogen. Remember, starch is another energy storage polysaccharide. It's just the version that is found in plants instead of animals. The individual glucose monomers or subunits are joined together, again in an alpha-1,4 glycosidic linkage. Like we saw in glycogen, the strands of glucose are going to form helices. But we see something slightly different in starch, and that is sometimes there'll be helices that are unbranched. That's what they're showing us right here. And in other forms, the helices will have branches coming off of them. This time, instead of having a branch come off every 10 glucoses, the branches come off every 30 glucoses. So it's much less branched than glycogen is. It's much less highly branched. And there are some helices that are completely unbranched. On this slide, we're looking at the first part of a table that compares these polysaccharides. This table comes out of a textbook, and I included it because it's a nice way to draw comparisons between the structures and the functions of these different polysaccharides. So in this first part of the table, we're comparing starch and glycogen. The names of the polysaccharides are listed on the left-hand side of the table, as well as where those polysaccharides are found and what their purpose is. Then we get to see the chemical structure in terms of the individual subunits, glucose for these, and the type of glycosidic linkage that joins them. And finally, we get to look at the three-dimensional structure of these polysaccharides and whether or not they are branched. Cellulose, remember, is not for energy storage. Plant cells produce cellulose because they need a polysaccharide that's going to give structural support to their cell wall. Cellulose is similar to starch and glycogen in the sense that it's built from lots of glucose molecules. But this time, the glucose molecules are joined together differently. They're joined with a beta-1,4 glycosidic linkage. Remember, this type of glycosidic linkage is much harder to break, and that makes sense because plants don't need to get at these glucose monomers. They put them into cellulose and keep them there to provide structural support. Now, one of the interesting things about cellulose is that the long strands of glucose do not form helices. Instead, they lie rather flat. And that's because within the strand, every other glucose molecule is flipped 180 degrees. By doing that, it causes the long chain of glucose to lie more flat and not get into that helical formation. So if we look at this portion of our summary table, we can see we're talking about cellulose here. We can see that cellulose is made out of individual units of glucose. This time they're bound together with beta-1,4 glycosidic linkages. And this time, if you go from one glucose to the next, you're going to see that it is flipped over 180 degrees. If you look at this first glucose, for example, you can see this, the portion that juts off of the ring, CH2OH, is oriented upwards, if you will, in this drawing. And if we go to the next glucose, it's going to be oriented in the other direction. So by flipping every other glucose molecule, we create long, flat strands. Now, what joins these parallel strands of glucose are hydrogen bonds. And that's what they've drawn in here for us. We're going to see hydrogen bonds forming in between the individual strands of glucose. Chitin is a polysaccharide that is used by insects, fungi, certain kinds of algae, and crustaceans as structural support. As I said earlier, chitin makes that hard exoskeleton around insects. It makes the shell around crustaceans, and it's found in the cell walls of certain types of fungi and algae. Chitin is an unusual molecule. It is not made out of glucose. Instead, it's made out of a simple carbohydrate, a monosaccharide, called NAG. Now, this is an abbreviation, but we don't need to know the entire name of this molecule. Everyone refers to it as NAG, so we will as well. The individual subunits of NAG are joined together with beta-1,4 glycosidic linkages. And similar to what we saw in cellulose, every other NAG is going to be flipped 180 degrees. That's going to allow chitin to assemble as long parallel strands. And those parallel strands will hydrogen bond to each other, just like what we saw in cellulose. Peptidoglycan is the most unusual of these polysaccharides. And that's because it has an unusual amount of strength within it. Of all the structural polysaccharides, peptidoglycan is the strongest. It's known to resist all different kinds of forces, pulling forces, pushing forces, and even twisting or torquing forces. Inside peptidoglycan, we find two types of monomers. We find two monosaccharides called NAG and NAM. These individual monosaccharides will alternate in the long parallel strands that make up peptidoglycan. Now, both NAG and NAM are fairly unusual monosaccharides. NAM is particularly unusual because it has a side chain coming off of it that's composed of a short chain of amino acids. When parallel strands of NAG and NAM line up next to each other in peptidoglycan, those amino acid side chains coming off of the NAM are going to sort of bump into each other. I'll show you an image of this in a minute. And what happens is peptide bonds will form, joining the amino acid side chains together. And in other words, joining the parallel strands together. So unlike what we saw in cellulose and chitin, where hydrogen bonds held the parallel strands together, in peptidoglycan, it's actually covalent peptide bonds that do this. This makes peptidoglycan very, very strong. In this last portion of the table that we've been examining, we can compare chitin and peptidoglycan. In chitin, remember, we see one type of monosaccharide repeating over and over again. That monosaccharide is called NAG. Every other NAG in the long parallel strands is flipped 180 degrees. That allows these strands to lay flat. When we compare peptidoglycan, we see both NAG and another kind of monosaccharide called NAM. NAM actually has a side chain of amino acids on it. That's what these green blocks represent. So we're going to build long parallel strands with alternating molecules of NAG and NAM. If you look at chitin, these parallel strands will be held together with hydrogen bonds. That's what the yellow dotted lines represent. In peptidoglycan, we see something very different. Everywhere that there are NAM in the alternating strands, there are these short amino acid chains. They're going to come in contact with each other, and they're going to form a peptide bond, one to the other. This allows for the parallel strands in peptidoglycan to be covalently bound to each other, making this polysaccharide very, very strong. On this slide, we're looking at several very common artificial sweeteners. And I put this slide in because I want us to take a look at these molecules and compare them to the natural carbohydrates that we see in cells. This molecule right here is an artificial sweetener called saccharin. This one is called steviol, goes by the brand name stevia. This is what aspartame looks like, very common in varieties of diet soda. And this last one is sucralose. It goes by the brand name Splenda. Artificial sweeteners have become very, very popular in our culture. They're used by people who want to avoid consuming natural sugar. And that's both to control blood sugar levels in some people and also to avoid the calories that are associated with natural sugar. When we consume these artificial sweeteners, our body can't break them apart, can't digest them, so they have 0 calories for us. They pass out of our body unchanged. Now, I asked a question on the lower left-hand side of the slide. Why are these molecules calorie-free? Why is it that our body can't break the covalent bonds in saccharin or stevia or any of the other artificial sweeteners? The answer is that our body does not have the enzymes that are necessary to do this. These are molecules that don't occur in nature. Some of them are based on natural molecules, but they've had certain chemical alterations made to them in the laboratory, and that makes them molecules that our bodies cannot break down because we simply do not have the enzymes that would be needed to break apart those particular covalent bonds. When you look at these artificial sweeteners and you think about what we've been talking about in terms of the structure of carbohydrate, the first thing that strikes you is that none of these, with the exception of sucralose, even resembles a carbohydrate. You don't see the ring structure in these that we typically see in a molecule like sucrose or fructose. If you look at saccharin, for example, we do have a ring structure here, but it's not a typical 6-sided ring. It's a 6-sided and a 5-sided ring side by side. And in addition to the carbons, hydrogens, and oxygens that we associate with carbohydrate, suddenly we have nitrogen and sodium in this molecule. Steviol is an even larger ringed structure. It actually contains 3 individual rings, which makes it different from what we would consider natural carbohydrates to look like. Aspartame really doesn't look anything like a natural carbohydrate. What's interesting is that this molecule was modeled after an amino acid, believe it or not. And finally, we have sucralose, which of all of them at least bears resemblance to carbohydrate. When you glance at this portion of the molecule, it looks similar to glucose or galactose or fructose. This linkage in the middle looks very similar to us. And even this at first glance looks similar. But when you look more closely, you realize that this is a 5-sided ring, not a 6-sided ring. But what makes sucralose unusual structurally is that in addition to the carbons and the hydrogens and the oxygens, we also have chloride. Chloride appears in several locations in this molecule. So it's interesting to note that artificial sweeteners cannot be digested because we simply don't have the enzymes necessary to digest them. And it's interesting to note that they have structural changes that make them quite different from naturally occurring carbohydrate. And finally, it's interesting to note that despite these differences, these are molecules that are able to bind to the same receptors that sweet carbohydrates bind to and signal to our brain that we're consuming something sweet. We're going to switch gears now and start discussing the lipids. Among the 4 macromolecules, the lipids are unique because unlike the carbohydrates and protein and nucleic acid, there's no repeating subunit in lipid. The other thing we're going to see is that lipid structure varies tremendously from one type to another. So no monomer to polymer relationship with lipids. There's no repeating monomer building a polymer. We see that in protein, nucleic acid, and carbohydrate, but not in lipids. Among the lipids, there are three major types. There are fats, or what are also called triglycerides. There are phospholipids. Those are the lipids that make up our membranes. And there are sterols. Each of these three types of lipid plays different roles in cells. Let's begin with fats. Again, fats are one type of lipid. It's important to remember that while all fats are lipids, not all lipids are fats. So we cannot use those two terms interchangeably. The purpose of fats in a cell is energy storage, specifically the long-term storage of energy. The kind of energy storage that carbohydrates play, starch and glycogen, for example, is short-term energy storage. For example, in animal cells, glycogen is used and depleted within 12 to 24 hours. Fats are different. Cells build fats and tuck them away for a rainy day. They're not intended to be used until they are absolutely needed. Over on the right-hand side of the slide, you can see a drawing of the chemical structure of fat. Up at the top of the molecule, we see glycerol. Glycerol is a 3-carbon molecule, and each one of those carbons has a hydroxyl group attached to it. Remember, these hydroxyl groups, these functional groups, are reactive. And when a fat is being built, it's built using a dehydration reaction, just like what we saw with carbohydrate. That hydroxy group is going to contact this molecule, which is called a fatty acid. All a fatty acid is a long chain of carbons and hydrogens. That's what this zigzagged line represents. And at one end, there will be a carboxyl group. This portion of the carboxyl group, the OH, will contact the hydroxyl group on the carbon, and a molecule of water will be released. So this fatty acid will be covalently bound then to the glycerol with a type of bond called an ester linkage. Now, one fatty acid can attach to this carbon, another can attach to this carbon, and a third to this carbon using dehydration reactions. So, all a fat is, structurally speaking, is a molecule of glycerol attached to 3 fatty acids. On this slide, you can see that fully formed fat right here. Here's the glycerol. Here are the 3 fatty acids. Remember, the long portion of the fatty acid is just repeating units of carbon and hydrogen. The chain itself is built out of carbon, and there are hydrogen atoms bound to each one of those carbons. At one end, there's a carboxyl group, and it's the carboxyl group that will react with the hydroxyl group in the glycerol, releasing a molecule of water and building an ester linkage. So each fat contains 3 of these ester linkages. On the right-hand side of the drawing, we're simply looking at a fat drawn a slightly different way. It uses this stick and ball diagram instead. Everywhere you see a black circle, that represents a carbon atom. Everywhere you see a white circle, that represents a hydrogen atom. And the red represent oxygen. It's important to remember that there's no repeating subunit in here. Fats are not polymers, and fatty acids are not the monomer, the repeating unit monomer. Fats provide long-term energy storage for cells. It's actually this hydrocarbon portion of the molecule that contains most of this energy. Fats contain 9 kilocalories of energy per gram. Remember, what we call calories are actually kilocalories. If you compare the kilocalories in a gram of fat to what's in a gram of carbohydrate, it's more than double. There are 4 kilocalories per gram of carbohydrate, 9 per gram of fat. Now, these fatty acids that are part of the structure of fats, they're going to vary tremendously in their length. Remember, we also see fatty acids in phospholipids. We refer to them as tails in phospholipids. The length of the tails in phospholipids can also vary tremendously. It's actually the length of the hydrocarbon portion of the fatty acid that is going to vary from one fat to another. The other thing that differentiates one fat from another is how saturated the fatty acids are. Remember, when we talk about saturation in lipids, we're talking about hydrogen. When we say that a fatty acid is saturated, it's saturated with hydrogen. That means that in that long chain, that long hydrocarbon, there are only single bonds between the carbon atoms. In an unsaturated fat, or a polyunsaturated fat, there are some double bonds between the individual carbon atoms. So a saturated fat has no carbon-to-carbon double bonds in the hydrocarbon portion of the molecule. The lack of double bonding allows for that fatty acid tail to hang quite straight, quite linear in its conformation. That allows those fatty acid tails that are coming off of the glycerol to pack together relatively tightly. And that makes saturated fat less fluid than other kinds of fat. We can see this in some of the fats that we enjoy eating. Think about butter. Butter is a fat that is saturated. And butter exists as a solid at room temperature. There are no carbon-to-carbon double bonds in the individual fat molecules. The fatty acids hang straight, the tails hang straight, and they pack tightly together, making this fat less fluid, solid at room temperature. If you compare that to, say, olive oil, which is an unsaturated fat, we see a much more fluid type of structure. Fats like olive oil are unsaturated, which means they contain carbon-to-carbon double bonds. Anytime you put a carbon-to-carbon double bond into a fatty acid, you're going to cause the fatty acid to bend or kink. That's going to make the fatty acid tails hang not straight, but bent and kinked. And that's going to open up physical space in that region of the molecule. So, saturated fats, no double bonds, less fluid. Unsaturated, carbon-to-carbon double bonds are present in the tails, lots of open space, more fluid. On this slide, down at the bottom, we're looking at a drawing of different types of fats. And we're looking at the distinction in the structure of the tails. Over here on the left, the blue circle represents the glycerol, and the black lines represent the 3 fatty acids, again, head and 3 tails. This represents a saturated fat. There are no carbon-to-carbon double bonds in these fatty acid tails, so they hang straight, they're linear. If you compare that to what we see over here, here we have unsaturated fats. This one would be considered a mono unsaturated fat, and this one would be polyunsaturated. Here's that glycerol head, 3 fatty acids. 2 of the fatty acids are hanging straight, but one of them is bent or kinked. That kink is created by a carbon-to-carbon double bond. Over here, we see polyunsaturated fat because there isn't just one carbon-to-carbon double bond. There are a couple of them. So there are a couple of bends, a couple of kinks. Those bends or kinks prevent the fatty acids from packing together. We get open space in between them, physical space in between them. These fats are going to be more fluid than this saturated fat will be. The second kind of lipid is the phospholipid, and we've looked at these previously. Phospholipids, remember, are the types of lipids that build up our plasma membranes, our cell membranes. Phospholipids have some similarity to fats in terms of their structure. If you look at this diagram, these diagrams, and focus in the center first, we still see a glycerol molecule, a 3-carbon glycerol molecule, and we still see fatty acids attached to it. But this time there are only 2. There are only 2 instead of the 3 that we see in fat. The other difference is that there's a phosphate group attached to the glycerol. All phospholipids have a phosphate group. This particular phospholipid also has another molecule attached to the phosphate group. I don't want you to worry about this right now. It turns out that different types of phospholipid can have all kinds of accessory molecules attached, but all phospholipids have a glycerol, a phosphate, and 2 fatty acids. Phospholipids are one of the macromolecules that are said to be amphipathic. Remember, when we use the word amphipathic, it translates as dual symphony, 2 natures within this molecule, both hydrophilic and hydrophobic chemistry. The head portion of a phospholipid is hydrophilic, and the tail portion is hydrophobic. Because of this dual nature, when you put phospholipids in a watery environment, they will spontaneously assemble themselves into a bilayer, with the heads facing the outside and the inside of the cell, where the water is, and the tails being tucked away in the middle. The head portion of a phospholipid is the glycerol and the phosphate, hydrophilic, water-loving. The tail portions, remember, fatty acids, these are hydrophobic. It's important to remember that a fatty acid is a hydrocarbon with a carboxyl group attached at one end. So the terms hydrocarbon and fatty acids, these are not interchangeable. Over here, on the left side, we're seeing a drawing of a phospholipid. Here's the head. We can see a glycerol, 3 carbons. And we can see 2 fatty acids hanging off. These are the tails. Attached to the glycerol is a phosphate group. So glycerol and phosphate make up the head. That's the hydrophilic portion of the molecule. That's the polar part of the molecule. The 2 fatty acids make up the tails. That's the non-polar portion of the molecule. Over here on the right, you can see a group of phospholipids, and they have spontaneously assembled themselves into a bilayer. This is how our cell membranes are built. The head portions on each layer are facing the water. If this were the inside of the cell and this were the outside of the cell, everywhere there's water, the heads are exposed. The polar heads of phospholipids will hydrogen bond with water. In the middle of that bilayer, that membrane, are all the tails. This is the hydrophobic part of a membrane. This is the non-polar part of a membrane. It needs to be kept away from the water. On this slide, you're simply seeing a different drawing of this same type of structure. You can see 2 layers of phospholipids drawn here. So we're looking at a portion of a cell membrane. The heads are facing the water here. If this is the outside of the cell, the extracellular space, the heads are all facing the water, interacting with the water, hydrogen bonding with the water. We see the same thing happening on the other side of the membrane, where there's also water inside the cell. In the center of the membrane are all the tails. These are hydrophobic. These are non-polar. They don't interact with water, but they will interact with each other, forming van der Waals interactions. It's van der Waals interactions, then, that hold the tail portion of a membrane together. It's important to remember that the individual phospholipids in a cell membrane are not covalently bound one to the other. They are simply weakly attracted to each other. The heads are held together with hydrogen bonding. The tails are held together with van der Waals interactions. This allows cell membranes to remain very fluid and flexible. The last type of lipid is the sterol. Sterols are also a component of cell membranes, but they also function as precursor molecules. Sterols are the precursor molecules that help cells build certain types of hormones, including the reproductive hormones. Sterols are a subgroup of another kind of molecule called a steroid. So the terms sterol and steroid are not interchangeable. All sterols are steroids, but not all steroids are sterols. What makes the structure of a sterol unique is this portion right here, drawn in orange. There's a large, bulky ring structure at one end of every sterol, and it's attached to a single hydroxyl group. So you can see there's a 6-carbon ring and another, and then a 5-carbon ring. And then there's going to be some other portion of the molecule that varies depending on the sterol. The most common type of sterol that's found in our cells and the cells of other animals is a molecule called cholesterol. Cholesterol is actually a critical molecule for the health of cells. We tend to think about cholesterol as being a bad thing because if we eat too much cholesterol, it can build up in the walls of our blood vessels and lead to cardiovascular disease. But the truth is, cholesterol is an essential molecule for living cells. Again, it's an important component in our cell membranes, and it's also a precursor molecule for a variety of different hormones, including the reproductive hormones. So cholesterol is used to build testosterone and estrogen and progesterone. Over here on the right-hand side of the slide, we can see the structure of cholesterol. Like all sterols, it has this big, bulky ring structure. 6, 6, 6, and 5. This is a large, bulky molecule. Remember, all sterols have this single hydroxyl group attached to this outer ring. When cholesterol is added to a cell membrane, it's added in the tail region. Because sterols are lipids, they are hydrophobic, and they will insert within the tails. What happens when a membrane gets more and more cholesterol added to it is the membrane becomes stiffer, less fluid. And that's because it physically occupies a lot of space. If you look at this drawing, you can see the heads of a group of phospholipids. Those are drawn in pink, and the tails are drawn in green. We said the tails sort of hang off of the heads, and if we're looking at a saturated membrane, the tails are all tightly packed together. Now let's start adding cholesterol in here. These yellow molecules represent cholesterol, and you can see it sort of tucks in between the tails. It pushes the tails out of the way, in fact, because it's such a big, bulky molecule. When you add cholesterol to the tail region of a membrane, you take up a lot of the space that was in the tail region. Less space means that these tails are more tightly packed together, and this membrane is less fluid. It's stiffer.